introduce Melanie Alvarado. She's defending her master's thesis work here. She's the first graduate student in my lab. She actually started here. She's an undergraduate at UAA, and she started research as an undergraduate research project in 2013. And she came into knowing that we had just started, and we're saying we're virologists, and she says, I want to work on HIV. I'm like, well, we're a flu lab. Let's see what we can do. So she took a look at virus-host interactions, so innate immune pathways that are common to many, many different viruses. She happened to look in the condition of latent HIV infection, and this contributes into a whole field of trying to cure HIV or come up with a functional cure for HIV, and she'll tell you a lot about that. So then she started she's decided to pursue this research as a master's degree, starting in 2014, and she's went through a lot of technical development of the entire system, started HIV research in our lab basically, how to use flow cytometry to do it, with a lot of help from some of you here as well. And some of the other students who have been working on this are here, so I'm really happy to see the next generation of students that will follow Melanie. Okay, so you want to take it away?
Sure. So thank you for all coming. My project is defining molecular pathways in the transcriptional reactivation of latently infected HIV T-cell lines. So what is HIV? It's a human immunodeficiency virus. It's an RNA virus, retrovirus. It facilitates the hyperactivation of the innate immune response, and eventually it will decrease T-cell production within the patient. There's roughly 35 million people living with HIV as of today, and 1.5 million deaths have occurred from HIV since 2013. Okay. So what's happening in the infected patient? This top black line is our CD4 T-cell count, and this red line is the HIV viral load within the patient. So starting at the acute phase, 4 to 8 weeks, you get a sharp increase in viral loads, and at the same time, you get a sharp decrease in CD4 T-cell count. Then for about 12 to 14 years, depending on how virulent the strain is, you have this chronic phase where that latent population, that population that's hidden within the patient that can't be detected, will actually become activated just slightly. Like let's say you get a cold or you get the flu during this time, it will waken up some of those cells and have a reaction. And at the same time, your CD4 T-cell count is trying to fight off this infection of HIV as well as any other infections it's encountering until about 2 to 3 years after this chronic phase, your CD4 T-cell count totally depletes and the virus takes over where the individual will die. Okay, so looking at the life cycle of HIV, starting with number 1, the GP120 protein is a surface protein on HIV. It binds to the CD4 receptor on the cell, and in order to enter the cell, it must bind to a co-receptor, CCR5. This allows it to fuse with the cell membrane, and then releases this capsid with the RNA into the cytoplasm. The capsid then dissolves, releasing the viral RNA, integrase, and reverse transcriptase. From there, the reverse transcriptase will turn the viral RNA into cDNA, and then integrase will shuttle the cDNA into the nucleus where it is integrated into the host cell's DNA. So from there, 1 or 2 things can happen. First, the cell can continue to produce viral transcripts where it will shuttle out and create TAT, REV, and NEF, essential proteins for producing infectious HIV. And REV will actually come back into the nucleus and help translocate large messenger RNA into the cytoplasm to produce like GAG and flow. After all of these proteins are produced, they migrate to the cell membrane where it starts to bud off and eventually will mature into an infectious virion. So from there, the virus can continue to infect other healthy cells. So the major portion of my project is looking at HIV latency, the other option that HIV can go into. And basically what this means is that after it is integrated into the host cell's DNA, it can revert back into silent state. So it looks exactly like a normal healthy T cell, but it's actually harboring the HIV virus. And that's why there's still no cure for HIV, is that these patients have these small populations of cell lines that are carrying HIV, but undetectable to their healthy immune cells. Fortunately, in the 1990s, they produced a therapy to prolong the lifespan of these individuals living with HIV, and that's known as antiretroviral therapy. It includes many types of inhibitors, entry inhibitors, reverse transcriptase inhibitors, integrase and protease inhibitors. They are all capable of suppressing the viral replication. Unfortunately, these individuals are still susceptible to secondary comorbidities. And what I mean by that is that an HIV positive person is more susceptible to cardiovascular disease compared to a healthy, normal individual such as you and I. And these drugs are very toxic to the patients. They can cause lots of side effects, even like kidney or liver failure. So it's really important that we try and expose this hidden population within these patients in order to expand their life. All right, so what is the driving molecular mechanism for reactivating latently infected HIV T cells? All right, so don't get scared by the diagram. I'll go through it. Okay, so there's this novel technique within the HIV research world where it's called shock or kick and kill. And what I mean by this is that they're using either viral RNA or some type of drug, injecting it into patients, and they're trying to awaken that hidden reservoir within them so that their healthy immune cells can go and kill off the infected cells. So in particular, in my research, I focused on a double-stranded viral RNA mimic, so it's a fake type of viral RNA. It's called poly-IC, and there's two forms with different weights, low molecular weight as well as high molecular weight. It is sensed through TLR3, which is a receptor that is able to internalize this signal and indicate to the cell, oh, we are under attack, we need to wake up and produce an immune response. Poly-IC, double-stranded viral RNA, is sensed through TLR3 as well as RIGI and MDA5. Once it's sensed, this signal that's internalized will activate these transcription factors. In particular, poly-IC should activate IRF3. And it can also activate these other transcription factors, IRF7, as well as NFkB. And with the IRFs, there's at least 9 of them within each person. And they can either homodimerize, such as right here, or heterodimerize with each other to initiate an immune response. And NFkB is very important in initiating an inflammatory response within the cell once it detects some type of infection by producing cytokines and chemokines and basically awakening up the cell, hey, we are under attack, we need to fight back. And another really important concept with NFkB is that on the HIV LTR promoter, it has 2 binding sites. So it should be required for activating HIV within these latently infected cells. So on the side, I just wanted to look at this other drug, R848. It's not a synthetic viral RNA at all. It's a drug that antagonizes another toll-like receptor, 7 and 8. It also is activated by RIG-I and MDA5. It will go through and mainly activate IRF7, but it can also reinforce the other pathways activating NFkB as well as IRF3. So I chose this drug to take a look and see if I would get a synergistic effect with poly-IC, and maybe we'd get even more reactivation with the combination. So overall, mainly I'm looking at poly-IC low molecular weight, and if I do get reactivation in these latently infected T cells, I want to see which of these transcription factors are required for HIV reactivation. So whether it's NFkB, IRF3, or IRF7. Okay, my investigation, general hypothesis is viral RNA poly-IC will induce HIV reactivation within these GERCAT GFP integrated HIV reporter T cell lines. It's a mouthful. And then reactivation of HIV within these latently infected T cells is through the transcription factors NFkB, IRF3, and IRF7, which do induce a type 1 interferon response. And what I mean by that is that IRF3 particularly will activate interferon beta, and then IRF7 will activate interferon alpha, which further initiates an immune response to an infection and wakes up those hidden cells. So AIM-1 is testing reactivation in HIV using the viral RNA mimic poly-IC, and aim number 2 is to determine which of the 3 transcription factors are required for HIV reactivation. All right, just as an overview, the main questions I'm asking: can poly-IC be used to reactivate that hidden population harboring HIV? And can we use this mechanic understanding of this pathway to produce a novel shock and kill therapy? All right, so the methods used, I used 2 different cell lines. They're both the GERCAT T cell GFP reporter cell lines that have HIV integrated into them. JLAT 9.2s are full length. They basically are very similar to an infectious virus, except for they're missing 2 major proteins, M and NF, that allow it to infect. And then JLAT H2s is the second cell line that only has a smaller portion of HIV. It basically contains LTR, TAT, and another LTR. And what's really important, a big difference between these 2 cell lines, is that they're integrated at 2 different chromosomes. JLAT 9.2s are integrated at chromosome 19, and JLAT H2s are integrated at chromosome 7. And again, on that LTR promoter, we do know there are 2 binding sites for NFkB, one of the transcription factors I will be looking at. So in order to observe if I did get HIV reactivation or green fluorescent protein, GFP, I used a Olympus microscope as well as flow cytometry techniques. And then, in order to determine whether these transcription factors were required for HIV reactivation, I used a technique called RNA interference that uses small interfering RNA to basically deplete those proteins within the cell. And I'm able to measure that with Western blot or RT-PCR analysis. Okay, so results for AIM-1, testing reactivation of HIV within these latently infected T cells using poly-IC. All right, so this graph is showing you JLAT H2s treated with a poly-IC low molecular weight at different dosages. And what's really great about this graph, this is RT-PCR. We're looking at relative gene expression on the y-axis, or transcript levels, and on the x-axis is my targeted genes I'm looking at after the treatment. So what's really great about this graph is that interferon beta, remember, it is activated by one of the transcription factors I am interested in, IRF3. And we are getting upregulation in a dosage-dependent response with interferon beta. So yes, all right. And then out of curiosity, I just wanted to see whether those toll-like receptors, TLR3 that senses poly-IC, and TLR7 that senses RA4A, whether I was getting an upregulation depending on the dosage as well. And then the other two genes are just activated later on after the innate immune response, the type 1 interferon, and I just threw those in there out of curiosity to see if they were activated as well. And they were, so yay. And then I put in this picture because I wanted to add in a funny story that we all know science doesn't work right away. And I was having so many problems trying to get the PCR machine to work, the primers, the protocol was not working. This graph took me 5 months to get. And so I was telling my friend about this, and she's like, okay, let's take a break, let's go to Vegas, go watch Pitbull one weekend. And I'm like, what? I should be working. She's like, come on, you need a break. I'm, okay, all right, I'll go with you. So we go and we're walking down the strip and we see this humongous lucky cat. It is the size of this wall. And she's like, go rub this cat's tummy. It'll help you with your PCR. And I'm like, okay, all right, we're in Vegas. And when I came back, I did this PCR again and I got this beautiful graph. So now I have a tiny one at my desk. All right. So this is looking at HIV reactivation in JLAT-H2s. And the main thing I want you to recognize in here is that each one of these glowing dots represents HIV reactivation within my JLAT-H2 T cell line. So this top A panel, we're looking at the control versus my medium concentration of poly-IC and my highest concentration of poly-IC low molecular weight. And as you can see, we do get quite a bit of an increase in green glowing cells, meaning that I am getting HIV reactivation visually. And then on the bottom, like I said, I wanted to experiment with the drug R848. So looking at it alone, after this is 48 hours, these are at 24 hours. 48 hours, you do get quite a bit of visual confirmation that green fluorescent protein HIV is being reactivated. And then the combination treatment shows quite a bit more green fluorescent protein HIV reactivation. So we wanted to be able to quantify this. How much more HIV reactivation are we seeing in poly-IC versus the combination treatment? So we used flow cytometry in order to quantify HIV reactivation. And just to go over what flow cytometry is, I borrowed this idea from Brenna Simons. So picture a gumball machine. You have a whole bunch of different colors, and picture each one of those colors being a different cell type. For instance, B cells are purple, and natural killer cells are red, and my T cells that are glowing green are green gumballs. So in my particular population, I'm looking at whether the cell is glowing green or not. So let's say the ones that aren't being activated are white gumballs. So I put it through the machine, it sucks it up, and a laser goes through each one of these cells and measures whether it's glowing green or not. And then we go to analysis, and this big blue population is my non-activated cells. And then within this box, each one of these dots represents a green glowing cell. And that's how we're able to measure HIV reactivation using flow. All right, so just a little bit about how I'm removing background from my samples. So I want to make sure that I'm getting legitimate results with poly-IC treatment and it's not just like spontaneous reactivation. So this top panel is showing you GERCATs. Those are the parental T cell line for JLAT-H2s as well as JLAT 9.2s. They have no GFP, no HIV in them at all. So looking at this blue box again, we're seeing 0% green fluorescent protein, green glowing cells. which is excellent because there shouldn't be anything in there or else something went wrong. And then on the bottom, I'm looking at my JLAT-H2 T cells, and we have about 0.14% background. And so with each of my samples, I would divide 0.14 under like poly-IC, even the control samples, in order to remove that background. And this is actually how I chose where to put the gate as well. And in the JLAT 9.2s, there is barely any background being produced with no treatment whatsoever. So I had to move this over 2 times 10 to the 3. Okay, so again, we're looking at flow cytometry, whether HIV reactivation is induced when I treat the cells with poly-IC low molecular weight, as well as high molecular weight. So looking at this graph, my treatments are on the x-axis. The y-axis is fold change. That just means if we're looking at the red bars, it's about like a 4 times higher activation rate than the control with poly-IC low molecular weight. So what I'd like you guys to take from this graph is that we did get significant increase in HIV reactivation when cells were treated with poly-IC low molecular weight as well as high molecular weight. And then bryostatin, this blue bar, is our positive control that specifically targets NFkB for activation. And that is also successfully inducing HIV. And let's see, combination treatment is at 48 hours, so poly-IC with R848, and it does show significant reactivation as well. When comparing the combination treatment with poly-IC low and high molecular weight, there was no significant difference. So this is really good to know, meaning that we can just use poly-IC within patients and not treat them with R848 because they're already taking so many drugs. It's best to use the smallest amount of treatment possible to reactivate this latently infective reservoir. And R848 did not initiate a very good HIV reactivation at 24 or 48 hours on its own. Okay, so this is looking at HIV reactivation in JLAT 9.2s, that full-length cell line. It's exactly the same thing. We're looking at flow cytometry versus the treatments, except for these cells are treated for 48 hours compared to the JLATs, which reactivate in 24 hours. Again, we are seeing significant reactivation when treating the cells with poly-IC high and low molecular weight. Unfortunately, the bryostatin did not show a significant increase, and this is most likely due to the low concentration we had to use in these cells, because later on I am specifically targeting NFkB for degradation, and it needs to be treated with a positive control. And they just were dying off with these high concentrations of bryostatin and Bay11. And so we most likely need to look at another, a less toxic NFkB inducer in combination with the Bay11, which I'll speak about later. All right, so A1, did poly-IC reactivate HIV in latently affected T cells? Yes, woohoo! So we observed GFP HIV reactivation visually, so we did see green glowing cells. In JLAT H2s, we got significant reactivation with both poly-IC low and high molecular weight, as well as the positive control bryostatin 1 and the combination treatment. And then in JLAT 9.2s, we did see significant reactivation in both low and high molecular weight poly-IC. So moving on to aim number 2, determining which transcription factors are required for HIV reactivation, including IRF3, IRF7, as well as NFkB. So in order to answer this question, I had to verify the knockdown or depletion of IRF3 and IRF7 within these T cells. So I did this one of 2 ways. First, with Western blot, measuring the protein using densitometry. And these Western blots have no treatments whatsoever. I'm only targeting IRF3 and IRF7. There's no poly-IC, no R848 at all with these cells. So I just want to find out the optimal time point for depleting the proteins. And if you take a look at the graph, the blue bars are IRF7, the orange bars are IRF3, and we do get quite a bit of depletion at 48 hours. Okay, then looking at knockdown of only IRF3 by itself, remember there's no treatment, no poly-IC, no R848 is being applied to these cells. So I'm only looking for the optimal time point. And looking at the graph, 48 hours looked very good at removing or depleting the IRF3 from within the cells. Okay, and then I was having a rough time trying to get the Western blot to work for IRF7, so we moved into RT-PCR analysis, which measures the RNA within the cell. And we're looking at fold change of the transcript level or gene expression at 48 hours and 72 hours within the cell. These are untreated. I just want to find out the optimal time point. And at 72 hours, it seems to be completely removed. And then another really funny story, Ralph here is a lab tech within the lab, and he performed this RT-PCR for me while I was doing my thesis. And he was working on it, and all of a sudden I see something like fly out of the hood. And I'm like, what was that? And I turn around, and we both stare at the ground, and he's been working on this for like 2 hours. And the plate was totally upside down. And the only thing I could say was like, is that the whole plate? I mean, it doesn't break apart. So I'm like, oh my gosh. The poor guy, he had a really good attitude. He jumped right back in it. I'm like, take a break, do it tomorrow, it's fine. He's like, nope, I'm gonna get right back in there. And we got these results, so yeah. All right, so was knockdown established? Yes, IRF3 with the Western blot, IRF7 with the RT-PCR. And so the next step is to quantify by flow cytometry whether HIV reactivation is happening or not when I deplete these two transcription factors. So again, we're quantifying using flow cytometry HIV reactivation with poly-IC treatment when IRF3 and IRF7 are knocked down in these cells. Okay. So the x-axis is my treatments. JLAT H2s has the 2 time points, 24 hours, and combination treatments are for 48 hours. And again, we're looking at fold change on the y-axis. And what's really great about this graph is that it's showing you with poly-IC high molecular weight treatment, we are getting a significant decrease in HIV reactivation. And what that means is when I remove IRF3 from the cells, HIV is unable to be activated. It's unable to be awakened. And we didn't see significant decrease with the poly-IC low molecular weight, the red bars, but I believe with like 1 or 2 more experiments, we would be able to see a significant decrease in those as well. And recently I just read a paper that came out this year. They were using human microglial cells that showed exactly the same thing. They treated the cells with poly-IC low molecular weight, and they showed that IRF3 is necessary for HIV reactivation. So this is great. I'm showing this in a totally different cell line that is previously not shown. And combination treatments, there was not much of a difference, possibly because the protein level was able to recover that extra 24 hours. All right, and then this was looking at the knockdown of IRF7 within the cells, and I ran flow cytometry on this at 24 hours, 48 hours, and 72 hours. There was no difference, no significant difference between those time points as well as the non-target versus the targeted cells. And I forgot to say that the non-target is just a control siRNA we add to the cells just to add the same amount that we're doing to the knockdown, IRF7 or IRF3. And something that was brought up to my attention is I looked at RNA levels, not the protein levels, and 3 papers discussed that the protein levels within cells actually deplete for IRF7 at the max 24 hours. So I believe, I'm very confident in this result at 72 hours that there's more than half of the protein removed from the cell, and so HIV just really doesn't need IRF7 for reactivation. All right, and then looking at double knockdown. So this is targeting both IRF3 and IRF7 within the cell with poly-IC treatments, low and high molecular weight. Again, there is no significant difference between the control and the knockdown samples. And I believe what's happening here is that another paper showed that IRF1 is a really good inducer of HIV reactivation, and it has a binding site on the HIV LTR promoter with IRF7. Together they can heterodimerize and activate HIV. So I think what's happening is when I remove IRF7 from the cell, it upregulates the amount of IRF1 that is being produced and can take over and establish reactivation. Okay, so overall, were transcription factors IRF3 and IRF7 required for HIV reactivation? IRF3 is required for HIV reactivation when using poly-IC high molecular weight. And again, with 1 or 2 more experiments, I believe I can show that there's a significant decrease in HIV reactivation with poly-IC low molecular weight too. And then IRF7 is not required for HIV reactivation using the poly-IC treatments. Like I said before, I believe that the protein is being depleted within the cells, and so I'm pretty confident in my result that it's not required. But what about that last transcription factor, NFkB, one of the major inflammatory response transcription factors? All right, so first, again, I needed to confirm that I was getting knockdown or depletion of NFkB within the JLAT H2s and the 9.2s. So this is the JLAT 9.2s full length. Again, I looked at the protein levels with Western blot. This is a phosphorylated antibody, NFkB. And what I'd like you to take in is within this square, the Bay11 treatments are less than the poly-IC treatments by itself. And Bay11 is a drug that specifically targets NFkB and inhibits it from moving into the nucleus and activating. So it basically, Bay11 turns off NFkB. And like you can see with these graphs, they are quite a bit lower. So it seems to be working in the poly-IC treatments. Unfortunately, in my positive control, bryostatin specifically activates NFkB, and Bay11 should inhibit that quite a bit. We didn't see that with the Western blot, but like I said before in the beginning, I didn't get much reactivation in the JLAT 9.2s using bryostatin. So we wanted to do 2 more experiments to make sure that Bay11 is targeting NFkB for reduction. So the second experiment was running an RT-PCR and looking at NFkB dependent genes. So on the x-axis we have poly-IC by itself and then poly-IC with Bay11. And then on the y-axis we are looking at fold change in gene expression or transcript level. And IL-8 is a gene that is specifically dependent on NFkB. So if we target NFkB with Bay11, we should get less of it, and we are seeing that. So each one of these Bay11s is less than a not targeted control treatment. So I looked at a different gene as well, MXB, and I didn't see quite the same thing. We weren't getting a depletion with the Bay11 treatment, and most likely this is because MXB relies on not only NFkB, but IRF3 and IRF7 within those cells. So we decided to do a 3rd experiment just to make sure that Bay11 is specifically targeting NFkB for degradation. All right, so this experiment was performed by an undergraduate, Jessica Kester, and she used an epithelial origin A549 cell line that had a NF-kappa B luciferase reporter. She used, on the x-axis, she has her treatments. Y-axis is looking at the amount of NF-kappa B being produced after the treatment. She used two different positive controls, prostatin and bryostatin. Both of them activate NF-kappa B specifically. And then she also looked at poly-IC low molecular weight. And as you guys can see, with the Bay11 treatments, she is getting reduction. So we decided to, did Bay11 inhibit NF-kappa B in the JLIT 9.2s? Yes, overall, Western blot showed a decrease in the phosphorylated NF-kappa B with Bay11 treatments. RT-PCR showed that IL-8 was reduced with the Bay11, and then the NF-kappa B luciferase assay performed in A549s, a totally different cell line, also demonstrated that Bay11 targets NF-kappa B. So, back to the original question, is HIV reactivation, when treated with poly-IC, dependent on NF-kappa B? So again, we're looking at flow cytometry. My orange treatments are the ones that have Bay11, that are targeting NF-kappa B and inhibiting it. Again, we're looking at fold change, and this is in JLAT H2s over 24 hours. And there is a significant reduction in the bryostatin Bay11 treatment. Unfortunately, we did not see significant reduction in the poly-IC treatments, but if you look at the overall trend, there is a decrease with the Bay11 treatments. I think with higher concentrations of the Bay11, we would be able to see a significant decrease in HIV reactivation. But unfortunately, the higher concentrations killed off the cells. I did a dosage response, and the highest I could use with the other treatments was about 3 micromolar. So we're definitely going to have to look at finding a different NF-kappa B target that's less toxic to these cells. Okay, and this is looking at the response in JLAT 9.2s when NF-kappa B is inhibited by Bay11. So again, we did see significant differences between the bryostatin treatments, but I wouldn't take this to heart. As I said before, the bryostatin did not reactivate very well within the JLAT 9.2s, and I couldn't increase the concentrations because of how toxic the Bay11 is to these cells. But again, overall, we are seeing like a slight decrease in the poly-IC treatments. So for AIM2, our transcription factors, IRF3, IRF7, NF-kappa B required for HIV reactivation when you use poly-IC. IRF3 was found to be required. IRF7 is not required for HIV reactivation, and NF-kappa B data suggests that it is required for HIV reactivation when you use poly-IC treatments. All right. So in conclusion, JLAT H2s and 9.2s require the transcription factor IRF3 to induce HIV reactivation when you're using poly-IC, high and low molecular weight. 1 or 2 more experiments would show that. And then, based on the Bay11, which targets NF-kappa B, treatments with poly-IC, the trend does suggest that NF-kappa B is required for HIV reactivation. But like I said before, we will have to find a less toxic NF-kappa B inhibitor. And then IRF7 is not required for HIV reactivation with poly-IC treatments. All right, future research. Like I said again, using a different NF-kappa B inhibitor, such as another nuclear translocation inhibitor, or targeting a different part of that pathway, possibly a P65 acetylation inhibitor. And then another experiment that could be performed would be knocking down both IRF1 and IRF7 As I said before, I think what happens when I knock down IRF7 is that IRF1 gets upregulated and it's able to maintain HIV reactivation. So if we knock down both of those transcription factors and we get a large decrease in HIV reactivation, then I've shown that IRF1 is actually taking the place of IRF7. And then eventually we will look at primary infected cell models and then move into patient donor cells. All right, so overall, antiretroviral therapy does not cure HIV. These individuals are susceptible to secondary comorbidities such as cardiovascular disease, and some of the drugs that they're taking, like Apcavar, is contributing to heart failure. And the protease inhibitors, they contribute to neurocognitive disorders such as memory loss. And these guys are still susceptible to like non-AIDS related cancers, and kidney and liver failure can occur from taking all of these drugs. So poly-IC is currently in clinical trials. It does induce type 1 interferon response in latently affected populations, but my results are showing you that poly-IC is specifically inducing IRF3, which is needed for HIV reactivation. So these findings can contribute to improving current therapies by producing specifically targeted combination treatments. For instance, now that we know that poly-IC specifically helps in activating that latently affected population, we can pair it with bryostatin, for instance, that specifically targets NFkB. That way you're not just throwing in a bunch of stuff and causing this person all these side effects and trying to awaken that hidden population. All right, so I'd like to thank my advisor, Eric Bortz. Thank you so much for letting me work in the lab and all your guidance, troubleshooting experiments. And my graduate committee, Kat and Jocelyn, for all your help in editing the talks and the thesis. And I'd really like to thank UAA Biomedical Group. Without their help, Brenna, Holly, Alex, I would not be able to get the flow cytometry data needed. And a lot of the undergraduates, Priscilla, created the dosage response for R848. William produced the bryostatin concentration. And then Ralph helped a lot with my RT-PCR. Jessica for her nucleophosmin or NFkB luciferase assay. And then, of course, to Carrie, who mentored me throughout my master's, and my awesome boyfriend, Ugo, and one of my closest friends, Mackenzie, and yeah, my parents, Brenda and Carlos Alvarado. So, great.
Questions?
Yeah.
If I got it correctly, you had a knockdown of IRF3 and 7.
Yes.
And where the knockdown of IRF3 on its own activated
Yeah.
the HIV provirus,
Yeah.
latent virus, and when you knocked down both,
Yeah.
that was no longer the case. Why do you think that is?
That's where I think the IRF1 actually comes into play. So I think when I knock down the IRF7, it actually takes over and it's activating in the place of IRF7. And so it doesn't really matter that the IRF3 is there or not. So I think IRF1 could actually induce HIV reactivation in the place of both of them. But
I see.
yeah.
But is it known to interact with IRF3 the way it does with IRF7?
No, actually, I haven't seen that. No. Yeah. But that could be looked into. That'd be a good future one as well.
Yeah, I've got a technical question.
Sure.
In your flow cytometry fold change figures, you have error bars associated with those means, which suggests there's replication of independent samples. And could you describe what was being replicated there?
Yeah, so those are two individual experiments. So each time I have a 12-volt plate, I ran the experiment. I did poly-IC treatments for 24 hours for 48 hours. Then I would go do flow, and then I'd start all over. I'd have the samples grown, treatment, and flow cytometry. Yeah.
On your early experiments with the time course treatment for poly-IC and the R848, you had poly-IC treatment at 24 hours, the R848 at 48 hours. I had to write this down because I was like, 48 hours. And then when you did the combination, you only showed data at 48 hours in combination. Did you actually look at earlier time points in combination?
No, so I decided to do that time point because I looked at R848 by itself, and at 24 hours, there was like nothing. There was no reactivation. And so at 48 hours, I was getting at quite a bit of green glowing cells. And so I decided to, at 24 hours of R848 treatment, I would add the poly-IC for the next 24 hours. And that's why I decided to do that.
Do you think if you treated it earlier, in combination at earlier time points, you might actually see a change in the response because of synergistic effects that you don't see on the individual basis?
There might be, but I was more concerned about killing off the cells. I think that combination would have been pretty toxic, and so I thought, 24 hours with one would not be too bad, and then you introduce the other, which is known to reactivate in 24 hours, and so that's how I decided. But that would be very good to look at, both of them at the same time. Yeah.
Kind of following up on Chris's stuff, which pathways are getting hit. So on the R848 treatments, the IRF3 knockdown, did you do R848 only in those IRF3 knockdowns?
Yes. Yeah.
Was that, did I miss it? Was it what happened there?
So with R848, I wasn't getting much of reactivation anyways, and so you can't really tell whether the knockdown even made an effect because R848 wasn't even induced by itself.
But then when you had the combo treatment, you did get still?
So I did get, with the combination, I did get reactivation in the first graph. But combination with the knockdown did not show any difference. And that's most likely that I think the protein is recovering because I have to add it another 24 hours.
Right, and is that possible? So that's one explanation. Could it be also like a different pathway, like IRF1 or something involved there?
Actually, it could be because that TLR7 goes through MYD88, and that's the same activator that induces IRF1. And so that could be a possibility, but overall, I would recommend not using R848 in HIV patients because another paper described how it actually suppresses HIV. So, and we also saw that in my reactivation, it was less compared to the poly-IC treatments by itself. It seemed to hinder the poly-IC in that combination. Yeah?
I guess I have a question about the toxicity of the bryostatin and the Bay11
Yeah.
that are affecting the NFkB. Have you actually checked with relatively normal cells what higher doses of those two drugs do? Because, I mean, it may not necessarily be a bad thing, right, if it were only toxic to your HIV-containing cells.
Oh, yes. Yeah.
And I wondered if you had actually tried either of those on relatively normal cells without HIV around.
No, that's pretty interesting, though. Yeah, no, I never tried that. Yeah. That would be good to look at. Yeah, jerk that's. Yeah.
Okay. So, these strategies are targeted to reactivate HIV.
Mm-hmm.
Taking a little step back, what impact are would they, especially poly-IC, which is a very generic compound in terms of immune activation, do you have to be concerned about reactivation of other latent viruses like CMV, herpes,
Oh, that's a good
in
question.
general, other?
Yeah, I guess it could actually do that too. So, yeah, I never really thought about that. It could activate those.
Yeah, that's my concern too. You're activating viruses. Now, the whole idea of the 5th and 6th generation HIV identification kits that came out is that we would knock down that viral load right away so we wouldn't have possible transmission of HIV. In fact, that's one of the keys to, you know, the fact that it's expensive to catch it early. It's better to catch it early when the virus is actually active so we're not spreading it and so we're reducing our overall cost and infectious agent. But you're talking about reactivating viruses, and all I can think of is, do we have to have them in isolation? I mean, I'm not saying that that's not a good idea. It's a whole idea of shock and kill, but I mean, even NIH is looking at the fact that the risk benefit might not be there, and they're still, you know, having an emphasis in research of just keeping it at that low unreplication level instead of trying to reactivate and kill because the risk is just too high.
Right, yeah, they were looking at treatments. Well, overall, the goal is to have a cure that eliminates it, but they are also looking at ways to maintain, like, what are they called? Controllers.
Right.
So yeah, and that's more of a healthy alternative as well, so that they are able to control it themselves without the antiretroviral. So yeah.
Right.
But I guess this is a treatment for the meantime.
It's kind of risky.
Yeah.
Lots of questions?